Forget what you thought you knew about how our cells recycle fats. Seriously.
Mm-hmm.
The long-standing idea was that this key lipid in our cellular cleanup crew, called BMP, was somehow untouchable, like indestructible.
Right, that was the dogma for a long time.
But hold on to your hats, because new research has unearthed the specific enzyme, PLA2G15, that actively breaks it down.
Yeah, actively chops it up.
This totally unexpected discovery could revolutionize how we understand and maybe even treat some really nasty diseases.
Okay, so just to set the stage a bit, our cells have these incredible recycling centers, lysosomes.
That's the garbage disposals.
Exactly, think of them like that. They break down all sorts of cellular waste, including complex fats, lipids. Now, inside these lysosomes, there's this special lipid bis monoacylglycerophosphate BMP for short, thankfully.
Much easier.
Way easier. And this BMP is super important. It helps other enzymes in the lysosome work properly.
Okay.
And we know its levels are messed up. They go haywire in lots of diseases, especially neurodegenerative ones.
Right, the brain diseases.
Yeah. So we recently figured out how BMP is made, but the million-dollar question has always been, how does it get broken down?
And the old answer was?
For decades, the consensus was just that its structure, its unusual chemical structure, protected it, made it resistant to breakdown. Full stop.
So, okay, this research team basically said, hold on, is that really true? Is BMP actually resistant?
Pretty much. And their first experiments using just, ground up bits of brain and liver cells, lysocytes, they immediately threw a wrench in the works.
How so?
Well, what was really surprising was finding BMP chopping activity, BMP hydrolase activity in these extracts.
Okay.
And not just under one condition, but under both acidic and alkaline conditions. That right away suggested, maybe BMP isn't this invulnerable thing we thought. There must be some enzyme involved.
And then they looked specifically inside the lysosomes themselves, right? That must have been even clearer.
Absolutely. When they used extracts just from lysosomes, they found really efficient BMP breakdown. And the key thing, this activity worked best at a pH between 4 and 5.
Which is?
Exactly the acidic environment inside a working lysosome. It's the sweet spot for a class of enzymes called acid phospholipases.
Makes sense.
Yeah, and plus, when they added this drug, amiodarone, which is known to block some of these lysosomal phospholipases.
Let me guess. The breakdown slowed down.
Bingo! It reduced BMP hydrolysis. So that strongly pointed towards a specific enzyme inside the lysosome being the culprit.
Which brings us to this enzyme, PLA2G15. You mentioned it's a major player in lysosomes, known for breaking down various lipids.
That's right. It's got a reputation for broad phospholipid hydrolysis activity. So the researchers naturally asked, okay, could this be the guy? Could PLA2G15 be the enzyme breaking down BMP?
This is like the obvious next step.
Exactly. So they tested it directly. They got purified PLA2G15 enzyme and mixed it with lipid extracts from mouse lysosomes. And here's where it got really interesting. PLA2G15 did break down most of the glycerophospholipids in there. And crucially, this included most types of BMP.
Regardless of, the fatty acids attached.
Pretty much, yeah. Regardless of a cell chain length or saturation, most BMP species were getting hydrolyzed. But importantly, it didn't touch other lipids like sphingomyelin or triglycerides.
So it's
This shows specificity, yeah. It's not just chewing up everything randomly.
And they saw this in human cells too, right? In HEK293T cells.
Similar results there, yes. And they went even further. They used a synthesized version of SSBMP.
Which is the real deal, the physiological form found in our bodies.
Precisely, the one that actually matters in biology. And they showed definitively PLA2G15 could directly break that down too. They could actually see the breakdown products using thin layer chromatography.
Okay, so pretty convincing. But they double-checked.
Oh, yeah. They used a mutated version of PLA215, one that couldn't work the S1980 mutant and also known inhibitors, like that amiodarone again, and another called fosinopril. Completely abolished BMP hydrolysis, shut it down.
Wow.
And just to be thorough, they tested another lysosomal phospholipase, PLBD2.
Did nothing.
Showed no activity against BMP.
Okay, so the takeaway from this first part seems crystal clear. PLA2G15 is definitely a lysosomal enzyme that can break down BMP. Case closed.
Well, not quite. That's the twist, right? Because it didn't break down all the BMPs. Some seemed resistant.
Ah. So why would some BMP resist? What's different about it?
That's exactly what they wondered. It forced them to look much closer at the different features of BMP structure. The details.
Like what?
Things like the specific types of fatty acids attached, where they're attached on the glycerol backbone. There are different positions called SEN positions, like SN2 versus SN3, SN3 pin, and also the 3D shape, the stereochemistry. There are different forms like RR, SR, and the physiological SS.
Okay, lots of variables. So they started testing these, like how fast PLA2G15 worked on BMPs with different fatty acids.
Yep. And what's fascinating is PLA2G15 was actually quite efficient against these BMPs, comparable to how well it breaks down another lipid called phosphatidylglycerol.
So it's not inherently slow against DMP in general.
Yeah. No, not at all. They also noticed the breakdown happens in 2 steps, taking off one fatty acid than the other, and that second step, the second deacylation, was a bit slower.
Okay. What about the 3D shapes, the stereoisomers? Did PLA2G15 prefer one?
Interestingly, no. Not initially, anyway. In shorter experiments, it seemed to hydrolyze all 3 stereoisomers, RRS, RSS, pretty much equally well if the fatty acids were attached at certain positions, these primary ester positions.
Okay, but then they looked at where the fatty acids were attached, the positional isomers.
Exactly. So picture the BMP molecule. You can have the 2 fatty acids attached at the SN2 and SN2 spots, that's 2-on-2-ep-2 spots, that's 2-ep-2 spot. Or you can have them at the SN3 and SN3 spots, that's 3-BMP.
And does it matter which one?
Well, the 2N-from-4 form is thought to be the more common one, the more physiologically important one in our cells.
And that difference in positioning 2 and a 2 versus 3 and 3 Fent, that's where they saw the resistance.
Bingo! A striking difference. The 2N2NMP was much, much more resistant to breakdown by PLN2G15 compared to the 3-BMP.
Really?
Especially over longer time periods. Even when they used the whole mix of enzymes from lysosome extracts, that 2-as-2 form held strong. They realized that just looking at the very first breakdown product appearing could be misleading.
So you had to track the disappearance of the original BMP over time.
Precisely. So they did careful time-dependent analysis, tracking all the different isomers with PLA2G15.
And that confirmed it.
It confirmed that all those primary ester stereoisomers got broken down similarly and way faster than the 2N-A BMP. They even figured out that the tiny bit of 2N-A2-AMP breakdown they did see was likely due to a small contamination with a different isomer, the 2-letter word form.
Did they look at how it might bind to the enzyme?
Yeah, they did some computer modeling, docking experiments. It suggested PLA2G15 has a pocket that BMP can fit into, and it seems to use the same basic mechanism as it does for other lipids, involving its catalytic site.
Okay, and when they looked really closely at the reaction speeds, the kinetics, with these positional isomers,
the difference was stark. PLA2G15 had high activity against 3-foot BMP, but against 2-foot BMP. Virtually no activity that depended on how much 2-a-2-BMP you added. It just wasn't being processed effectively.
wow, so it's really about that 2-FBD position.
Well, almost. One final crucial experiment. They tested different 3D versions, different stereoisomers, but all of them had the fatty acids in that 2-a-2 efficient.
And what did that show?
That revealed the final piece. It's the combination of the 2-a-2 position and the specific SS stereochemistry, the natural form that provides the significant resistance.
So you need both.
You need both for maximum protection. If you had the 2-a-2 position but a different stereochemistry like RR or SR PLA2G15 could break those down much more easily.
Okay, so let me get this straight. The really key insight here is that it's the specific 2-on-2 placement of the fatty acids on the physiologically relevant SS form of BMP that makes it tough for PLA2D15 to chop out.
Exactly. It's not just the SS shape alone, which was the old idea. It's the shape plus that specific 2-on-2 linkage. That provides the resistance.
That is a major refinement of our understanding. Really highlights how subtle structural details matter in biology.
Absolutely. And it raises fascinating questions like, how do cells control this? Do they actively maintain the 2-in-2 OR?
Okay, so they've shown PLA2G15 can break down BMP, but the main form is resistant. Does PLA2G15 actually matter for BMP levels in a living cell or organism? Is it the physiological BMP hydrolase?
Right, that's the critical question. So they looked at cells where the gene for PLA2G15 was knocked out, meaning the cells couldn't make the enzyme.
And what happened to BMP levels?
In PLA2G15 deficient HEK293T cells and in the lysosomes isolated from them, most types of BMP significantly increased. They piled up.
Okay, that's strong evidence.
And it wasn't just one cell line. They saw it consistently across multiple cell types.
Could they reverse it? Like add the enzyme back?
Yes, they did a rescue experiment. They took lysosomes from the deficient cells and added back purified working PLA2G15.
And BMP levels went down.
They did. The accumulated BMP was reduced back towards normal levels. But interestingly, If they added back the inactive mutant PLA2G15,
Nothing happened?
nothing happened. BMP stayed high. It really showed the enzyme's activity was required. There is a little complexity, though some BMP species, especially those with a specific fatty acid called 22.6, weren't fully rescued, hinting there might be more nuances.
But the overall picture was clear, and
Yeah.
they confirmed this in other systems too.
Yep. They saw similar BMP accumulation in PLA2G15 deficient HeLa cells, and importantly, in multiple tissues, brain, kidney, liver, from mice that lack the PLA2G15 gene.
And crucially, other lipids weren't affected in the same way.
Right. Things like sphingomyelin levels stayed mostly unchanged. It pointed specifically to a problem with BMP turnover.
Could it be that BMP synthesis was increased somehow rather than breakdown being reduced?
Good question. They checked that. Levels of the enzyme that makes BMP CLN5 weren't elevated. Other lysosomal markers were normal too, and experiments suggested BMP synthesis rate wasn't increased either.
So it really points to reduced degradation.
Exactly. The most straightforward explanation is that losing PLA2G15 leads to BMP accumulation because it's not being broken down properly.
So PLA2G15 is key for VMP turnover. How does this play out dynamically inside the lysosome, and does it affect other lysosomal functions?
Right. They first confirmed, using pulse chase experiments, that BMP degradation was slower in lysosomes from the PLA2G15 deficient cells.
Okay.
Then they did clever experiments using cells that naturally have low BMP levels, CLN5 knockout cells, and delivered different BMP isomers to track their fate. This confirmed that the resistant 2LO2-BMP was indeed the major form found inside cells.
And showed that PLA2G15 speeds up its breakdown.
Well, indirectly, they showed that the breakdown of this delivered 2LO2-BMP actually requires it to first rearrange, to undergo something called a sil migration, shifting a fatty acid to become the 2-ven-3 or 3-laurin-300 isomer.
Oh, so it has to become the non-resistant form first.
Exactly. And those forms, especially the 3-3 isomer, were very rapidly degraded, particularly when active PLA2G15 was present.
So PLA2G15 mops up the non-resistant forms quickly, which pulls the equilibrium away from the resistant 2-2X form over time.
That seems to be the mechanism. They confirmed this by adding mixtures of 2-L2-over and 3-L-3-L-over BMP. The 3L8 got chewed up fast, while the 2Le2-over remained largely untouched directly by PLA2G15.
Yeah.
Its degradation relies on that prior rearrangement.
What fascinating interplay. Does messing with PLA2G15 affect other things lysosomes do?
Yes. Since BMP is known to help another important lysosomal enzyme, glucocerebrosidase, or GCase, work better.
Okay.
They found that knocking down PLA2G15 actually increased GCase activity. Makes sense, right? More BMP hanging around, more GCase boost.
Right, more helper lipid.
Exactly. And inhibiting PLA215 macrophages had the same effect, while adding extra PLA2G15 decreased GCase activity.
Wow, so PLA2G15 levels can tune other lysosomal activities via BMP.
Seems like it.
Yeah.
And this led to a potentially therapeutic angle. PLA2G15 popped up as a hit in a genetic screen looking for factors that could influence Niemann-Pick disease type C1 or NPC1.
NPC1, that's a lysosomal storage disease where cholesterol builds up, right?
Precisely. And they found that knocking down PLA2G15 actually reduced that toxic cholesterol accumulation in patient fibroblasts.
So less PLA2G15 leads to more BMP, which somehow helps clear cholesterol in this disease model.
That appears to be the connection. It suggests PLA2G15 influences overall lysosomal lipid balance, and inhibiting it might actually be beneficial in certain disease contexts.
Okay, reducing cholesterol buildup in cells is one thing, but does getting rid of PLA2G15 actually help in a whole animal model of NPC1 disease?
That was the crucial next step, testing the therapeutic potential in a living organism. They used NPC1 deficient mice, which model the human disease.
And they knocked out the PLA2G15 gene in these mice as well.
Exactly, creating double knockout mice, and the results were pretty remarkable.
How so?
Key disease biomarkers in the cerebrospinal fluid and plasma, things like nerve filament light chain, which indicates nerve damage, and a STALT, indicating liver damage, were significantly improved in the double knockout mice.
This is suggesting less neurodegeneration and liver problems.
Correct. Interestingly, the BMP levels themselves only showed minor changes in these mice. It might be due to cell type specific effects or perhaps the overall reduction in lysosomal stress, lessening the impact on BMP.
But other storage lipids were affected.
Yes. Other lipids that typically accumulate in NPC1, like certain sphingolipids and another one called alkylylysophosphatidylcholine, were significantly reduced in the brain and liver of the mice lacking both NPC1 and PLA2G15.
What about the actual pathology? Did the brains and organs look better?
They did. Neuropathology showed reduced loss of Purkinje cells in the cerebellum, less inflammation markers like astrocytosis and microgliosis, and less demyelination in the central nervous system.
Significant improvements across the board there. And other organs.
Visceral organs also showed benefits. Less Kupffer cell hyperplasia in the liver, reduced issues in the spleen. The mice lacking only PLA2G15 showed no lesions at all, by the way.
So getting rid of PLA2D15 seems pretty safe on its own. Did the MPC1 mice function better? Did they live longer?
Yes, huge functional improvements. They performed better on tests of neurological function and motor coordination. And critically, getting rid of PLA2G15 significantly extended the lifespan of the NPC1 deficient mice.
Wow, that's a really strong result.
It really is. It strongly suggests that targeting PLA2G15 offers genuine therapeutic benefits, at least in this mouse model of NPC1.
So let's wrap up this deep dive. Thinking about the bigger picture, what are the truly innovative aspects here, and where does this research point us next?
Well, I think the absolute standout innovation is just completely overturning that decades-old belief about BMP being inherently resistant to breakdown. That's huge.
Right, shifting the paradigm.
Exactly.
Yeah.
And then identifying PLA2G15 as a key enzyme that does hydrolyze it, and figuring out the structural twist that it's the 2-3-1-2 up position with the SS stereochemistry conferring resistance. That's groundbreaking stuff.
It really changes how we have to think about BMP metabolism.
Completely. It adds layers of regulation we didn't know existed, involving enzyme activity and potentially acyl migration.
And looking forward, what new questions or research avenues does this open up?
Oh, plenty. A big one is figuring out what the unidentified biological machinery, as the paper calls it, might be that protects the 2-out-2-month BMP from rearranging into the more easily degraded forms. How is that stability maintained?
Yeah, how does the cell control that acyl migration?
Right. And related to that, how do cells maybe preferentially make or preserve the resistant 2-out-2 main form? Then there's that little quirk with the 22.6 containing BMPs in the deficient models that needs more investigation. And of course, the most exciting avenue perhaps is the therapeutic one. Developing specific targeted inhibitors for PLA2G15 seems like a really promising strategy now, not just for NPC1, but potentially for other lysosomal storage disorders or neurodegenerative diseases where we know BMP levels are off.
So investigating PLA2G15's role in those other diseases is key too.
Absolutely. This discovery has really reset the landscape and given us a whole new set of tools and questions to explore lysosomal function and disease.
So just to quickly recap for everyone listening, the really remarkable finding here is that BMP, this crucial lipid in our cellular waste disposal system, the lysosome, isn't actually indestructible like we thought.
Yep, not at all.
There's an enzyme, PLA2G15, that actively breaks it down, and it turns out the specific way the fatty acids are attached to the BMP molecule is the key to its relative resistance.
And understanding this really intricate dance of lipid breakdown, it doesn't just deepen our knowledge of fundamental cell biology. It also opens up really exciting possibilities for treating diseases like Niemann-Pick type C, which are currently very hard to manage.
Yeah, it gives researchers a concrete target, PLA2G15, to go after for potential therapies.
Exactly.
Which leaves us with a final thought to chew on. If inhibiting PLA2G15 can have such dramatic benefits in NPC1 disease by tweaking BMP levels, what other cellular processes, what other diseases might be influenced by fine-tuning BMP through this enzyme?
Right. And are there other molecules inside our cells, things we currently think of as stable, that might also be under unexpected enzymatic control like this?
It really invites us to question those long-held assumptions and just keep exploring the incredibly dynamic nature of what's going on inside our cells.